Hello everyone, Mr. Linder here. Let's talk about digestive secretions. So it turns out that we secrete about 7 liters of fluid into our GI tract daily. And now those secretions are going to be things such as acids, bicarbonate, bile, mucus, digestive enzymes, salts, and of course water into our digestive lumen. Roughly half of the secretions come from GI tract accessory organs, so things like salivary glands, pancreas, the liver. The rest of the secretions are going to come from epithelial cells that line the GI tract mucosa. A large portion of the fluid that is dumped into the GI system is going to be water and ions. The ions that we secrete are going to be things like sodium, potassium, chlorine, bicarbonate, hydrogen ions. So there's a lot of ions that are being moved into the digestive lumen. When you move ions into the digestive lumen, you can also move water as well. So water is going to follow the ions based on osmotic gradients. But it really depends on which direction that they're going. So if you're secreting ions into the digestive lumen, then you're moving water into the luminal space, which is essentially the outside of the body. If you were to move ions back into the epithelial cells and then, of course, into the lymphatic system or cardiovascular system, then you're going to be reabsorbing water back into the body and not losing it to the outside. And so there has to be that homeostatic balance between the way that water moves. It turns out that the epithelial cells have a variety of membrane proteins that allow us to move various types of ions. So when you look at particular diagrams, like for example this one here, you're going to notice that there are a variety of pumps that are found in the epithelial cells. This one here is a hydrogen potassium pump. There are chlorine channels. These are CFTR channels. And so depending upon what types of proteins are found in the epithelial cells, that's going to dictate what kind of secretion a particular cell will have. So let's start with the idea of acid secretion in the stomach. So acid secretion is accomplished by parietal cells that are part of the gastric glands of the stomach. So if you were to look at the enlargement of the mucosa of the stomach, what you're going to see is you're going to see what's called a gastric gland. And the gastric gland is where you have a variety of cells, for example, G cells and D cells, and these parietal cells, enterochromaffin-like cells, etc. So there's lots of different cells in the gastric glands. But it's in particular right now the parietal cells that we're interested in looking at the idea of acid secretion in the stomach. So it turns out that the parietal cells will produce anywhere from about 1 to 3 liters of secretion per day. And they can create a pH as low as 1. Typically we see, though, in gastric secretions about a pH of 2, but it can get as acidic as 1. What's interesting, though, is that the pH in the parietal cells, so pH inside the cytoplasm, is about 7.2. So it turns out you have a high concentration. So we have a high concentration of hydrogen ions in the luminal space of the stomach, and we have a low concentration of hydrogen ions in the parietal cells. And so anytime you move something from low to high concentration, if we're going to be moving ions into the lumen of the stomach and you're moving them from low to high, then you're going to have to be able to pump those things. So you have to have a pump, which really implies that you're using energy. Right? You have to pump those ions from a low concentration to a high concentration. So you're going against the concentration gradient. So when you look at this idea of secretion of hydrogen ions, really you're looking at a way to pump those hydrogen ions into the gastric lumen. So where do the hydrogen ions actually come from so that we can actually create an acidic environment in the lumen of the stomach? So I want to take you to a different diagram here. So let's go to this one. On this particular diagram, what I like about this one is it shows you the equation for where the hydrogen ions are coming from. So this is our parietal cell that we're looking at on the screen. So I'll just put P for parietal cell. So this is the parietal cell in the epithelial membrane, in the mucosa of your stomach. And cells generate carbon dioxide from metabolism. So CO2 is a waste product from metabolism as cells are generating ATP. So this cell is essentially, if we write over here, this cell is taking glucose and it's running it through metabolism in the purpose of producing energy. The cell needs ATP. And in that process, many steps of glycolysis, the transition reaction, the Krebs cycle, the electron transport chain. But as this cell is generating energy, you are producing carbon dioxide as a byproduct. So that carbon dioxide is mixing in water. So it's produced, carbon dioxide is produced by metabolism. It's mixing in the water that's in the cytosol of the cell, and it's being enzymatically catalyzed into carbonic acid. And so here we have the enzyme labeled. So here's carbon dioxide plus water, enzymatically catalyzed. So this is carbonic anhydrase. Carbonic anhydrase is the name of the enzyme that is going to catalyze this reaction. Ends in ACE, so we know it's an enzyme. And so we generate carbonic acid, H2CO3. Carbonic acid then can dissociate into hydrogen ions. So here's the hydrogen ions that we're producing in the parietal cell, and bicarbonate ions. And so we get bicarbonate and we get hydrogen ions from the dissociation of carbonic acid. The hydrogen is then used by this pump. So here we have a hydrogen potassium pump, and I'm going to write ATP here because it's utilizing energy in order to pump against the concentration gradient. So this hydrogen is diffusing through the cytosol, and then it's making its way to this protein pump. And now we have the hydrogen leaving the cell, and so it's going into the gastric lumen. And as that hydrogen enters the gastric lumen, it is making the lumen acidic. Okay, more hydrogen ions is going to create more of an acidity in the luminal space of the stomach. In exchange for that hydrogen, we have potassium coming into the cell. So this is what we refer to as an antiport. When one ion goes one direction and one ion goes the other direction, this is an antiport that is taking place. And it's a primary active transporter because it's using ATP directly to move hydrogen from a low concentration to a high concentration. What happens to the bicarbonate ions? The bicarbonate ions are moved into the cardiovascular system. So they're moving out of the parietal cell towards the cardiovascular system in exchange for chlorine coming into the parietal cell. So this too is an antiport, but it does not require ATP. So this is simply utilizing the concentration gradients for bicarbonate and chlorine. So this is an exchange that's taking place on the basal side of the parietal cell. That chlorine will diffuse through the parietal cell. So we have diffusion of the chlorine moving through the parietal cell, and then that chlorine will be transported out into the luminal space as well. So we see the chlorine leaving the parietal cell and entering the gastric lumen. So bicarbonate is entering, in essence, bicarbonate is entering the blood, and that actually can act as a buffer in circulation. What we notice is that the blood that is leaving the stomach actually has a slightly more alkaline pH than normal blood. So we refer to this as an alkaline tide, if you will. So there's alkalinity as the blood is leaving the stomach. And then the chlorine that has traveled through the cytoplasm is entering into the gastric lumen. And so the net here is the production of hydrochloric acid. But it's really just hydrogen ions and chloride ion. So that's your HCl, or hydrochloric acid, that is produced in your gastric secretions. Another type of secretion to take a look at is bicarbonate secretion that we see coming from the pancreas. The pancreas is actually producing bicarbonate, and that bicarbonate is moving into the small intestine. And the reason that the pancreas does this, this is the small intestine in the diagram right here. And so the pancreas is moving bicarbonate. So we have bicarbonate moving into the small intestine. And the reason there's a large amount of bicarbonate coming from the pancreas is because the stomach is releasing its gastric juices with digested material into your small intestine. So the digestive juices, this gastric juice, has a large concentration of hydrogen ions. We just saw how you can put those hydrogen ions into the stomach to make it acidic. So those hydrogen ions that are coming into the small intestine would make this environment very acidic. So we're going to buffer those hydrogen ions, and we're going to create an alkalinity. So here's the hydrogen that's coming in. We're going to bring the bicarbonate from the pancreas so that we can buffer this environment. That way the pH in the small intestine is going to be more of a value of 9. It's going to be much more alkaline. So you see a pH of 2 coming from the stomach, and yet we have a pH of 9 in the duodenum, in the small intestine. And that's because there's this bicarbonate secretion that is helping to neutralize the stomach acids. So bicarbonate coming from the pancreas, moving into the duodenum so that we can neutralize the stomach acids. So the vast majority of bicarbonate is being produced by the pancreas, and specifically it's being produced by the pancreatic duct cells. So these are the duct cells. If we were to zoom in on the pancreas, these are ductal cells, okay? DUCT, ductal cells. And so we can see that there's a bicarbonate production coming from those ductal cells. You also have acinar cells. The acinar cells are producing digestive enzymes, so that's these guys right here. Also in the pancreas, you have endocrine cells. So you have cells that are producing insulin and glucagon, but we're focusing on the ductal cells right now. We want to see secretion into this tube, into this pancreatic duct, and then we want to see that bicarbonate move along the pancreatic duct and be dumped into the small intestine. So how do these ductal cells produce the bicarbonate? Okay, so bicarbonate is coming from the ductal cells. And again, we're going to use carbonic anhydrase again, just like we saw in the parietal cells of the stomach. So what we have going on is the production of hydrogen and bicarbonate again. So on this particular diagram, what we're looking at is this is a ductal cell. So this is a ductal cell. And this is going to be the lumen for the pancreatic duct. So this is the pancreatic duct. And so what's happening, again, is that carbon dioxide from metabolism is mixing with water. And that CO2 and water is enzymatically catalyzed by carbonic anhydrase so that we can produce bicarbonate and hydrogen ions. Now remember, there is a carbonic acid. There's that H2CO3, and then we have this dissociation that occurs into hydrogen ions and bicarbonate. So this is very similar to what you see in a parietal cell. Only in this case, we're going to move the hydrogen and the bicarbonate in different directions. So this time, the bicarbonate is going to be moved into the lumen. Okay, so we see an antiport between bicarbonate and chlorine on the apical side of the cell. So this is the apical side. And then we're going to see the hydrogen ions moving out of the cell on the basal side. So it's the opposite of what we see in the parietal cells. So bicarbonate and hydrogen ions are produced when CO2 and water are enzymatically catalyzed by carbonic anhydrase. The bicarbonate is going to move into the pancreatic duct. Hydrogen ions are going to move towards the cardiovascular system. So how does this all come into play? Well, there has to be chlorine in the system. So we have chlorine that is transported by CFTR channels. So that chlorine is being moved into the lumen. And then notice that it's being exchanged through an antiport with the bicarbonate. So as bicarbonate goes out, the chlorine comes back in. And then that chlorine can then cycle back through the CFTR channel. So we get bicarbonate into the pancreatic duct by a bicarbonate chlorine antiport. The hydrogen ions are exiting the cell using an antiport with sodium. So here we have a sodium and hydrogen antiport where the hydrogen is going out and the sodium is coming into the cell. So we have a hydrogen and sodium antiport, and then the hydrogen is moving its way into the cardiovascular system. So that's actually going to make the blood from the pancreas a bit more acidic. So remember, the blood from the stomach is a bit more alkaline. The blood from the pancreas is a bit more acidic. So there's definitely a balancing that's taking place where you have alkalinity from the stomach and acidity from the pancreas, but that blood in the cardiovascular system is going to balance each other out. In order for the sodium to stay in a concentration gradient so that it can assist with this antiport, you have to have a sodium potassium pump. So the sodium potassium pump, we'll mark this with ATP so you know it's using energy. The sodium potassium pump is working to ensure that sodium keeps its high concentration. So this is going to give us a high sodium in the interstitial space, and that's going to allow that sodium to come back into the cell. You'll also notice that sodium is also coming in through this SYM port. Okay, so this is a SYM port right here. SYM port means that the ions all move in the same direction. So this is a sodium, chlorine, potassium SYM port. This is giving us the sodium that is necessary to be used by the sodium potassium pump to be pumped back out of the cell, and then the sodium can come back in exchange for the hydrogen, or it can come in through the SYM porter. So we have the sodium potassium pump always working, and we can essentially move some of that sodium across the cytosol. So some of that sodium that's coming in can actually move across the cytosol and move out into the lumen as well. We can get sodium out here in the lumen. We also get sodium that's moving paracellularly, so between the cells. Okay? So we get sodium moving this way. Okay? And so that's going to give us this sodium bicarbonate that is produced in the pancreatic duct. But again, it's the bicarbonate that we care the most about because that's going to be our buffer for the stomach acids that are coming into the duodenum. From a clinical standpoint, if these CFTR channels are not functioning properly, you may notice that they are labeled CF, which stands for cystic fibrosis. So these are cystic fibrosis transmembrane channels. If a genetic defect was inherited, then these chloride channels would not be functioning. And that's what you see in cystic fibrosis. So cystic fibrosis isn't just a respiratory condition where you see chloride channels affected in the respiratory system. It also affects the digestive system channels as well, and the pancreatic channels and so forth. So all chloride channels are being affected by this genetic defect. And so this results essentially when the chlorine is not transported properly, it's going to result in a thickening of mucus, and it's going to clog these ducts, and it's going to prevent buffers and digestive enzymes from properly reaching the intestines. And so that's going to be another symptom for cystic fibrosis patients having digestive issues. Another secretion to take a look at is simply salt secretion, so sodium chloride secretion. You know that when we move ions, we can also move water with it as well. So similar to pancreatic secretion, where you basically have this sort of isotonic saline solution that's being dumped into the GI tract. So we are essentially moving salts into the luminal spaces. So here we can see chlorine being transported into the luminal space of the GI tract. We get the sodium and water that's being transported as well. Where is this all coming from? What is powering this? Well, of course, it's being powered by the symport. That symport where we move sodium, chlorine, and potassium. That's going to move sodium into the cytoplasm of our epithelial cells. That's also moving the chlorine into the cytoplasm of our epithelial cells. We get the potassium that's moving in as well. We have potassium transporters that allow the potassium to re-exit. We also have our sodium-potassium pump that's always on and always functioning. So here we're dumping sodium back out of the cell. And we also have potassium coming back in, and that's our sodium-potassium pump. Sodium can move paracellularly. So it's moving between the cells and it's bringing water with it. Chlorine is being moved by our CFTR channels into the luminal space. So we're essentially getting sodium chloride, we're getting salt, we're getting water. And so that's creating a liquid balance in our GI tract. If we didn't properly move salts and water into the GI system, we would have constipation. The GI tube, the lumen, would get dried out. We'd have dehydration taking place, and that would ultimately lead to constipation. However, we do see situations where some bacteria can overly affect these channels. And so we see sometimes that chlorine and even sodium is moving too well into the lumen. It's drawing too much water. And, you know, if you have too much water, then that's going to lead to diarrhea. So things like cholera can cause diarrhea because it's affecting these chloride channels, and you're getting too many ions and too much liquid into the GI space. Other types of secretion that we see in the digestive system would be digestive enzyme secretion. Digestive enzymes, I mentioned earlier, are going to come from the pancreas. Digestive enzymes come from the stomach. This is an example of pepsin being produced by the stomach epithelial cells. Pepsin is a digestive enzyme that breaks down proteins. You have salivary amylase that's coming from your salivary glands. So there's a variety of salivary glands, parotid, sublingual, submandibular. So these are producing an enzyme called salivary amylase. That's, again, another type of secretion. So there's going to be a lot of secretion of digestive enzymes into the luminal space so that we can break down carbohydrates, proteins, lipids, nucleic acids. And so there'll be future videos on enzymes and all of their digestive functions, but lots of enzymes that are being secreted into the digestive lumen. We also have a lot of mucus secretion. So mucus secretion is going to come from specialized cells in the GI tract epithelium. Those are going to be essentially mucus neck cells, or essentially what we call goblet cells. So there's mucus neck cells in the stomach. There are basically serous type cells in our salivary glands, and of course what we call goblet cells throughout the epithelium of the GI tract. And so these cells are going to produce what are called mucin proteins. Mucin is going to mix with water, and it's going to form a viscous material that we refer to as mucus. Mucus coats the GI system, so it's protecting the GI tract. It's also lubricating the GI tract, and it's making it easier for material to move through the tract. This secretion is going to be controlled by the parasympathetic nervous system. It's going to be controlled by various types of cytokines and even neuropeptides controlling the mucus secretion in the GI tract. We also have, obviously, saliva production. So in that saliva, there's going to be the salivary amylase. Saliva is coming from these salivary glands. There's going to be mucus in the saliva, so water, ions, mucus, digestive enzymes, even antibodies in our saliva to protect us against bacteria or viruses that are entering through the oral cavity. And again, salivation is controlled by the autonomic nervous system, specifically the parasympathetic branch of the autonomic nervous system. One of the last secretions to take a look at would be bile secretion. Bile is produced by our liver. So in this particular diagram, we're looking at the liver up here. So bile is produced by the liver. Bile is stored in the gallbladder. And then we move that bile down through the bile duct. So this is the common bile duct. And so that bile is going to dump into the small intestine. So bile is a secretion that ends up in the duodenum. And we actually use bile for the emulsifying effects that we have for lipid digestion. The key components of bile, so if you are to look at where bile is coming from, so it's coming from the hepatocytes of the liver. It's being dumped into these bile ducts and it's collecting eventually into the gallbladder. But what are we looking at in terms of what's in bile and what do we use it for? So bile is made up of various bile salts. And those bile salts are important because these are the emulsifying agents. They have both a hydrophobic and hydrophilic end to them, and so they can orientate themselves into fat droplets and then help us to create smaller fat droplets so it's easier for fats to be digested. There are also bile pigments that are in the bile, for example, bilirubin. Bilirubin is a pigmented molecule. It comes from the metabolism of hemoglobin. You're also going to find cholesterol in bile. This is a way that we can actually eliminate some of our excess cholesterol in the body. We need cholesterol for plasma membranes, but in excess, we need an excretory route, so cholesterol ends up in our bile and can be eliminated in our feces. And then we also see a variety of drugs and other foreign molecules that end up in the bile for excretory reasons. So, for example, on here it's showing different types of antibiotics: ampicillin, streptomycin, tetracycline, penicillin. These are all essentially drugs or xenobiotic molecules that are going to end up being moved into the bile and then can be eliminated from the body through the GI system. So bile is created by the hepatic cells in the liver, and then it's eventually moved into the gallbladder for storage, and then we're going to release that bile through the common bile duct into the duodenum. And it's actually being controlled by the hepatopancreatic sphincter, and that is controlling the flow of bile and the bicarbonate that comes from the pancreatic duct. It's controlling that flow into the small intestine and allowing us to secrete these materials into the small intestine to use further in the digestive system. So that's a little bit on secretion in the digestive system. I hope that helps. Take care.